Methamphetamine, other known as crystal meth. Forensic Biology Bio 101 D80 by Alexandra Cicchetti. Let's learn some facts about crystal meth before we begin. It can be smoked, snorted, swallowed, or injected. If the meth is smoked, it's usually through glass pipes. If snorted, it is of course generally through the nose, and if injected, it is commonly inserted into the anus or urethra. It appears like shiny rocks or pieces of glass in various sizes. The drug itself is generally colorless, but sometimes has a blush pink tint to it, and is odorless, having no significant smell. The effect can last all the way up to 12 hours, which is much longer than most drugs with the similar effects. When used, crystal meth increases the dopamine level in the body, which interprets the body's pleasure. It is usually taken for euphoric effects, causing intensive excitement or happiness along with a great deal of energy. This drug has many nicknames. Some of them include meth, crank, chalk, or speed. Speed is the other name that you will commonly hear people refer to crystal meth as. This drug is strongly and highly addictive. Most people say they had become addicted to it even only after the first time that they had done it. The difference between synthetic and natural drugs. Synthetic drugs are designed to replicate organic botanical compounds, but they consist of extremely processed chemicals. The human body has trouble distinguishing synthetic drugs, making it more difficult to handle and metabolize them. Natural drugs or organic drugs are considered to be natural substances which are derived from plants and animals. The synthetic derivative of crystal meth. Crystal meth is a synthetic man-made chemical generally made up illegally in undisclosed laboratories. Keep in mind, amphetamine is another stimulant type of drug. Crystal meth is made by infusing numerous schemes of amphetamine or byproducts with other similar substances to advance its efficiency. Familiar pills used for cold medicine are usually served as the base for the creation of this drug. A meth producer infuses additives from those cold pills and elaborates the strength as it's combined with chemicals. Chemicals in this substance can include battery acid, drain cleaner, lantern fuel, and antifreeze, which cause for the intake to be highly dangerous. How crystal meth affects the central nervous system. The central nervous system is composed of the brain and spinal cord. It serves as the center of our body's actions, regulations, and maintenance of almost every single one of our bodily functions. Crystal meth, proven by research, mainly focuses on its effects of the brain, although there is still some effect to derive on neurons located in the spinal cord. Using increases neuronal death, which means crystal meth decreases the number of neurons throughout the central nervous system, which means in many scenarios cannot be removed. This stems from brain damage in numerous areas of the brain. The first effect on the central nervous system is decreased gliogenesis. This occurs mainly in the prefrontal cortex of the brain, where a great deal of human functions stem from. The prefrontal cortex controls one's attention, planning, abstract thinking, judgment, etc. Crystal meth use causes a decrease in the making of glioprogenitor cells. These tender cells serve into different types of glial cells. The second effect on the central nervous system is decreased white matter. Once the glial cells become damaged, the brain and the spinal cord's capability to yield myelin is then affected, causing white matter in the central nervous system to be decreased. White matter is what advocates active authorization between neurons in the central nervous system. Due to the location of these neurons, once the matter is decreased, neurons end up being less dynamic in transmitting their signals, stemming a great amount of functional deficiencies. The third effect on the central nervous system is increased glutamate calcium in the brain. This is the outcome of neurons becoming exceedingly excited in the brain and causes them to chronically pry, thus damaging or harming the system as a whole if it is not balanced. The fourth effect on the central nervous system is the neurotoxic effects that damage the dendrites of the neurons. The dendrites of neurons collect portions of the neurons that collect the chemical signals from other neurons. This causes damage by blocking those neurons from getting through completely, and in the end affects a large portion of the cognitive and motor functions. Crystal meth effects on other organs or tissues. Although the effects of crystal meth are most symbolic in the brain, it can also cause long-term damage primarily to the human heart, liver, lungs, and skin. Effects on the heart. In the heart, crystal meth can cause blood vessel spasms, rapid heart rate, high blood pressure, and the death of muscle tissue and the formation of fibrous tissue in the heart. This leads to shortness of breath, heart attacks, coronary artery disease, cardiomyopathy, and aortic dissection. Crystal meth effects on other organs or tissues continued. When doing crystal meth, one experiences an increase in libido, which is a craving to have sex, risking infectious diseases, which include HIV/AIDS, hepatitis B, and hepatitis C. Crystal meth diffuses the immune system, causing the body to become more susceptible to diseases. Oral health and skin complications are frequent with meth use, which is where the terms meth mouth and meth sores are known from. Generally, one experiences thinning or patching hair, tooth decay, cracked or dry skin, gum disease, sores, and weight loss. After a significant amount of time, these effects continue to worsen. Crystal meth in the society. Although crystal meth is not a new drug, in recent years its power has striked due to a spark in manufacturing facilities. Crystal meth in earlier years. In earlier years, crystal meth was much more compelling and simpler to make. Back then, it was commonly used in war as an injection to keep troops and soldiers awake. It is also used as a support aid for diets and depression. After so long of making matters worse for drug abuse, the government made the drug illegal for most uses. Crystal meth today. This striked its production and distribution to be made illegally in labs and sold illegally with no regards on what was affiliated with the drug. Today, crystal meth is generally used by individuals of all ages. It is most familiarly used as a club drug. Generally, it is taken while partying, whether at a nightclub, rave, etc. Some people also take it because it can lead to accelerated weight loss, but once one has stopped taking it, the weight comes right back. The United States Drug Classification. The USDA, which is other known as the United States Drug Enforcement Administration, classifies drugs into 5 specific categories. They classify them in schedules, which depend upon the acceptable medical use, the drug's abuse, or probable dependency of a specific drug. What class of drug is crystal meth? Crystal meth is an illegal street drug. This drug falls in the same class as many other street drugs such as cocaine. Due to the fact that the drug is man-made and does not use natural ingredients, it is classified as a synthetic drug. Crystal meth, methamphetamine, is classified under Schedule II. This means it is identified with a high rate for abuse, which is with a likely cause of serious psychological and physical dependence. It is a risky experiment because of all the new chemicals developed, which are constantly growing, having users with no way of accurately or positively knowing what the crystal meth that they may use will contain. How can crystal meth be used as a forensic tool? This is forensic drug chemistry. Crystal meth can be used as a forensic tool within crime scenes where a specific illegal substance may have been suspected to have some sort of connection to the crime committed. Questions can lead to the substance itself, containers that were used to bring the drug there, utensils that assisted in the manufacturing of the drug, or the fundamentals needed to create the drug. For situations like these, biological samples, blood, and urine can all be used as evidence found at the crime scene. This evidence is then collected, photographed, packaged, documented, and delivered for analysis in the process of conclusion. Weight tests, presumptive testing or screenings, and confirmatory tests are beneficial in scenarios like these. Each laboratory has their own specific procedure and evidence analysis, but these are general analysis procedures. And here are my references. Thank you.